Yes, thanks a lot, Ashil, for the nice introduction. I mean, usually I would thank the organizers for giving me the opportunity to talk. I mean, this time it's a bit different. I'm really excited to actually see so many people here in the attendee list. I mean, back when I had this idea and I shared it with Phil, Ashil, and Zolt, I mean, we didn't imagine, back then that this could be such a big event. But thanks to COVID now, and also thanks to Zoom, the time has changed, and I'm really excited, and I hope that you really like the talk so far, and that you will benefit also for the ones that will come in the afternoon session, and also the panel discussions. So I really hope that you can take something back from today's symposium. I mean, before I go into the daily business that we do in our lab, in our group, I just wanted to, mention something or give you an advice. So back when I started here at the hospital, there was no bioinformatics community, no network of all the bioinformaticians that are here at the hospital. And I think really my advice would be, get together in your hospital, in your institution, and really found a community and network, because this is really important. I mean, first of all, it gives you the opportunity to share ideas, to exchange, problems and so on, to find solutions among each other. And we do this usually like with a bi-weekly meeting. In our case, it's like every second Thursday that we meet, and it's a very informal meeting. And the nice thing is one of the products now that came out of this community effort here is this symposium. So I would definitely really advise you and urge you, to really get together and form a network of bioinformaticians in your institution. All right, so let me now dwell a little bit into what we do on a daily basis. So our lab, the Molecular Tumor Profiling Lab and the Clinical Bioinformatics group within this lab, has actually, I would say, 3 different fields or 3 different, task areas. One is the most important one is the diagnostics. So we do daily diagnostics for cancer patients, where we sequence their tumor, DNA to identify targeted therapies for precision medicine. But besides that, we also, because we have multiple sequences, so we regard ourselves also as a sequencing facility. And indeed, we really do whole genome sequencing, whole exome sequencing, RNA-seq, and so on, for not just single patients, but also for larger cohorts of hundreds and thousands of patients and individuals. So, and if you have any projects, feel free to get in touch with us. We are always excited about these projects, and please get in touch. And then the third field is basic research, and here my personal interest lies in alternative splicing and proteomics, and how this can be combined with the genomic data that we sequence in our lab, also again to foster precision medicine. But now, because the session here of the symposium is about the current daily routines and how bioinformatics can help in those daily routines. I want to just focus on the diagnostics part. And what I would like to do is I would like to introduce you to comprehensive genomic profiling assays, and also show you a little bit about how these can be applied in tumor boards, and what we came up with a solution, because it's not straightforward actually to use these genomic profiling assays at tumor boards due to the complexity. So let me just tell you a little bit about those comprehensive genomic profiling assays. Actually, you heard a little bit already about them from Valerie Barbi's talk. Basically, cancer is caused and is driven by by mutations in the genome, and they are, if you can categorize them, 4 different classes of mutations that are also clinically and therapeutically relevant, and these are base substitutions that cause mutations in or exchanges in amino acids in the protein that can then activate, deactivate the protein, the gene products. You have also insertions, deletions that lead to frame shifts, and very often tumor suppressor genes causes then the loss of the function of the tumor suppressor gene. Then you have copy number alterations where oncogenes are amplified, or again, tumor suppressor genes would be lost in their copy, and this again has impact on the cancer. On the cancer development, and then you have also rearrangements and fusion events. So where a gene gets fused to a different gene promoter, and or can be actually fused to an intergenic region where it again loses its function. So there are these 4 different categories of mutations, and with comprehensive genomic profiling assay, we are trying really to identify all 4 types of mutations in one single assay, so in one single experiment. And it's called comprehensive because we don't do this just for one or a couple of genes, but we really try to do this to as many genes as possible. And as Valerie pointed out, actually the genes that you are interested in are genes that are really therapeutically actionable. So we don't, right now for the clinical daily routine, we don't need exome sequencing because most of the genes in the genome are actually not actionable. So here with the 324 genes and the FoundationOne assay, we are really covering actually all, the large proportion, the majority of cancer-related genes, and we can identify all the mutations or most of the mutations basically in those cancer-related genes. And this list is actually already itself very large, and I'm just showing you here basically the table, the gene table that Foundation Medicine provides. And as you can appreciate, I'm pretty sure most of you will know some of the genes, but actually there will be no one that actually that knows all of the genes by heart. So, and this is actually one of the problems that comprehensive profiling assays bring on the table, where bioinformatics then really can help. Is just simply the sheer volume of data and information and genes, and then to assess which ones are actually which mutations are actually pathogenic and relevant for the patient in terms of treatment. And another very important aspect of comprehensive profiling assays is that they have to work on formalin fixed paraffin embedded samples. And I'm just showing you here, basically on the left-hand side, how a tissue block that is embedded in paraffin looks like. And what we usually do is we slice then a slide of this block, a couple of slices, and then extract the DNA from those slices and put them out. Do library preparation before we put them on the sequencer. So this is also very important that that the assay that you're using is really applicable to FFP material, because FFP material is the standard material still in the hospital and the clinical daily routine, and it's not like cell cultures or not fresh frozen biopsies. So really like still the standard are these formalin fixed paraffin embedded tissue blocks, and that's not easy because the samples are actually the cancer samples and the cancer cells undergo certain modifications and certain changes when they are fixed. So your kit or your assay has to be able to handle those. And one of the problems also that when you work with these tissue blocks is that you don't have always pure cancer cells in your tissue that you sequence. Very often, for example, we have occasions where we just have 10% of cancer cells in the tissue that we are sequencing, and the cancer sample that we are sequencing. On top of that, you also heard early on from Mark Rubin that cancers are quite heterogeneous. So sometimes you have subpopulations and so on. And in order to identify now mutations that are low frequent, you really have to sequence quite deep. And that's also the advantage of those targeted panels, that you can indeed sequence quite deep. And in our case, with the FoundationOne assay, so we sequence on a median 500x and very often over 1000x. And important is also that you have an even coverage of all the exons, because you don't want to lose a mutation or miss a mutation due to the fact that you haven't sequenced that exon that well. So in our case, we can guarantee that 95% of exons are covered at least 100x. And you see here on the left-hand side, so the coverage is in exonic regions and also in the intronic regions. And the intronic regions are very important because most of the fusion events and rearrangement events are happening in the intronic regions. And then the nice thing is, because now we are, because we sequence so broadly and we have actually quite large target size in our assay, we can actually do more than just identifying single mutations. We can also look for biomarkers. And here, the most important biomarkers are the tumor mutational burden and the microsatellite instability. So those are two biomarkers that are indicative of a large number of mutations in your genome. And if you have a large number of mutations, then actually your immune system and the T cells actually should be able to recognize the cancer cells, and due to that, a recognition should actually destroy the cancer cells. But because this is not happening, because you see the cancer cells, so there must be something that is blocking actually this this recognition. And for that, a new class of therapy, immunotherapies, have been developed. And actually, just a couple of years ago, there was a Nobel Prize awarded to those checkpoint inhibitors, to those immunotherapies. And you can see here in this Kaplan-Meier curve that, if you have a high mutational burden, so this is the blue curve, if you have lots of mutations in your genome, then actually giving immunotherapy to those patients really increases their overall survival by, substantially. And that is the reason why we are really looking for those biomarkers in all of our cancer patients to identify patients that might benefit from those immunotherapies. And there's another biomarker, loss of heterozygosity, which just Jan Christenat elaborated a little bit, which is, which indicates that you have homologous recombination deficiency. And this is also something that our panel can elaborate or can make a statement on. The thing is, with comprehensive profiling assays is actually the complexity is really a burden. And so the nice thing with the FoundationOne assay is that actually you have a nice report which tries to bring some structure into all of the results. And I'm just showing you here the report. So here you see on the left-hand side, so we have all the patient and ordering physician information on the top left, and then you have a summary of all the genomic alterations on the top right-hand side, and also, including the biomarkers that I just mentioned, so tumor mutational burden and the microsatellite instability. And then you have at the bottom, you have targeted therapies. So those are therapies that the patient could receive because he or she has mutations in his tumor for which targeted therapies were approved. And if you have a high tumor mutational burden, for example, you can even receive immunotherapy. And there are here a list of immunotherapies listed for the patients. And very important is also, in this case, the patient has options, but often the patients also don't have options due to the fact that drugs have not been approved yet. But very often, actually, in those cases, we have clinical trials, so we can actually provide a list of clinical trials where new therapies or new combination of drugs are tested, and where the patient could then enroll in those clinical trials. The problem with those reports is with comprehensive profiling assays is a lot of text. So I'm just showing you here, the selection of the pages. So this is a 24-page long a booklet, and now you can just imagine, like if you are just dealing with one patient, that's kind of okay. So you can, read this and try to understand it. But now just imagine, what will you do if you have to discuss 10 patients or 15 patients or 20 patients now at the molecular tumor board? This brings me now to the next topic, because at molecular tumor boards, actually what we have to ensure is that all patients that are discussed are really discussed thoroughly, and that we don't miss any potential therapy options. So what are molecular tumor boards? So molecular tumor boards are multidisciplinary meetings where the NGS results, for example, coming from a comprehensive profiling assay, are put on a table, and where then experts, in this case oncologists, pathologists, bioinformaticians, I really have to say here, bioinformaticians play a really important role in those tumor boards. Geneticists, molecular biologists are all coming together, and the data is put on a table, and then at the final end, a recommendation is written down in a tumor board report, which is then sent to the oncologist of the patient, so that he can then decide on the best therapy option. But the problem is, you can now imagine these comprehensive profiling assay, with the amount of data that they have, it's just really very hard to discuss more than one patient at the tumor board, and especially given that we have to discuss 10 cases per week. So in general, we have like 5 minutes per patient, and the data. And you don't want to waste, if you have just 5 minutes per patient, you don't want to waste 1 or 2 minutes just to get all the data together of the patient in order to discuss it. And what we did is now in our group is we developed a new software, basically, that really helps us to understand and to better, more efficiently discuss NGS data of patients. And I just forgot to mention this. So at the USZ, we really do this on a weekly basis. So we have a molecular tumor board every week happening. Usually, actually Thursdays. It will happen today at 2 o'clock. So this is really something that is absolutely daily routine. The software that we wrote now to discuss or to make NGS comprehensive profiling assay data readily available to a tumor board is called MTPpilot. So it's a pilot that helps you to dwell into the NGS results of a computational profiling assay. So how is this constructed? So the MTPpilot feeds from an MTPpilot database. That database is a Microsoft SQL database, and you can access that database via a web browser. And this web browser was written in PHP, HTML, and JavaScript. And actually, the MTPpilot database feeds itself from a set of other databases. And we have categorized them into 3 different. So we have fundamental databases where the underlying structure of the data relies or sits on. This is ensemble genomic data, cosmic cancer data, uniproteome data. And then we link actually mutations to different databases like NOMAD, ClinVar, OncoKB, ARUP, and so on. Also, Valerie talked a little bit about this. So, we do it very similar to that. And then we have annotation databases where we bring now additional stuff on the table, where we help interpret interactively the data. And we use data from PDB, string, SMART, gene ontology, and CAC pathway. And to that, we have to also add, obviously, patient information. So we connect to the hospital patient information database. Then if there are pathological data available, like H&E slides, images, or any other reports that are important, we can also link to those. And important is also that we take the mutations actually of the patient, obviously, and feed them all in. And important is also that we have a lot of outlinks. So for every mutation, for every patient, there are multiple databases to which we outlink, where you can then do further analysis and investigations. And I just want to show you now how the web browser looks like. So this is on the left-hand side, you see all the patients. So every week we can select from the MTPpilot, we select the week of for which we want to see now all the patients. And now you see on the left-hand side the patients, for example, for that week. So it is, in this case, the November last year. And in this case, it's anonymized. So usually we would have a patient name and with birth date and so on in it. And then you see on the top, you see the patient information. So when you click on the one of the patient, the page on the right-hand side changes, obviously. So you see then the patient information at the top. You have resources like the medical report, the F1 report at the top right-hand side, readily available. You can just click and then it will open the PDFs. You have the overview of all the mutations in an ideogram at the top, and you have then for every single mutation, lots of different annotations to help understand whether a mutation is really relevant or not. And I just want to quickly maybe go one by one through different functionalities here. This is a very important actually figure, the ideogram, which gives you immediately access to all the mutations that have been observed. You can immediately see, for example, now I got used now to it, so I really see it immediately. So I see all the single nucleotide variances. So these are in green circles, and I see the copy number alterations in those squares. So either it's amplified, in this case it's lost because it's blue, and I see also fusion events. And this is actually a prostate cancer patient, and Mark Rubin talked about this TEMPRAS fusion, and in this case, this is also a patient that has this TEMPRAS fusion event where actually the black line here, the cytoband has been removed, and two of the genomic regions now are fused together to form then a TEMPRAS ERK fusion event. Then we have a lot of, as I said, a lot of annotations readily available. And here, very important is, that we have that we can show, for example, whether a mutation is a hotspot mutation. Given that you have 300 genes, you don't know whether, what are the hotspot mutations for every single gene. But we now with bioinformatics, we can do that. So we have a tool that detects those hotspot mutations and will tell you, for example, in this case, that you are the second most frequent mutation that has been observed for the gene KRAS, for example. So we can immediately tell the clinician, yeah, this is a hotspot mutation, so it's a frequent one. And then what is also very important is that we know where the mutation is located in the domain structure of the protein. In this case, you see the p53 domain structure, and you see the red label here for the location of the mutation, and it's right exactly in the DNA binding domain of p53, which is one of the regions that is most frequently mutated. Then we have also the possibility to to visualize the mutation on protein structures. So you can even, if in this case the mutation has actually been observed in the protein structure itself, so you can then align the wild type sequence of the structure to see how what is the impact of the structure of the mutation on the structure. You can change the different visualization representations, and you can even select different protein structure altogether if you think that this is more useful. So this is something that is also a tool that we frequently use, and more frequent is actually here the fusion viewer. And the fusion viewer is really something quite crucial, where we can get to know which, where the breaking points are for the fusion events. So in this case, it's again this TEMPRAS fusion. So you can see the intron, the first intron has the breaking point for TEMPRAS, and the third intron has the breaking point for the ERK gene, and they are fused then together to form this gene fusion or fused gene here in the middle. And you see basically that all the protein domains here from TEMPRAS are lost due to that, to the fusion events, but actually that you have the protein domains from ERK actually still remaining in the fused gene. I mean, in this case, it's kind of a very well-known fusion event, but you can just imagine, if you can do this automatically for any fusion event. So you have all these data always readily available. And because it feeds from a database, this whole tool, you can also compare patients with each other based on the mutational profile. And the TEMPRAS fusion is a fusion event that is really very, is solely observed in prostate cancer patients. And this is what you can see here, that the TEMPRAS fusion is really something that is only observed in prostate cancer patients. And this patient match is also very important for us for cases, for example, where we don't have a diagnosis and where we actually would like to know which patients are most similar in terms of the mutational profile to the patient who doesn't have, a known cancer type. And what is also quite exciting is that we we have readily available the H&E slides. So these are the slides that I was talking about, where we cut off from the tissue blocks, and those are stained now. And you can just go on this, on the viewer and have a look at the, at the tissue. And this is also important for sometimes where we don't exactly, where we maybe are unsure with the diagnosis, and where we just would like to have the opinion of all the pathologists and physicians in the tumor board, what they are thinking about. All right, and there's actually much more. So we have a PDB viewer, a PDF viewer, obviously, for the reports. We can compare, cases. If a patient has been resequenced multiple times, we can compare those mutations with each other. There's a cancer type matcher, so where we can see whether the diagnosis and our mutations are fitting well with the cancer types in Cosmic. We do also along the line that Jan Christnet was explaining regarding LST. So we are also working on this so that we have an LST and copy number plot. And then we have IGV viewer, so you can look at the mutations in the raw data. We have string interaction networks of mutations, and also CAC pathways are also highlighted. And for the future, actually, we want to now have a CAC pathway matcher, so that you can really actually visualize the mutations on CAC pathways. We are working on mutational signatures to identify them from those smaller gene panels, which is also not straightforward, but we are working on it. In the future, we'd also like to do molecular modeling, especially for mutations where we don't exactly know whether they are pathogenic or not. And also like visualizations of clinical trials and therapy options. This is also something that we are right now working on. We want to ensure that no clinical trial is really missed at our tumor boards. And we're also excited that very soon we will actually be going online with a public version of the MTPpilot software. And I'm really looking forward to get, if you have any feedbacks in the future, please, try that tool out and let us know. With this, I would like to thank a lot of people. So first of all, the clinical bioinformatics group. So here in particular, Batur, Domingo, Jakob, Tulay, and Uli. And but actually, the front end of the MTPpilot software was written by Fabian Arnold, who's also part of our lab. And then a big thanks to the Department of Pathology and Oncology, and also to Foundation Medicine for their constant support, and also the Krebs Zurich for financial support. And if you have any questions or so, you can, this is my Twitter handle and my email address. Okay, and with this, I would like to thank and take questions if you have any.
Okay, brilliant. Thanks, Abdullah. Does anyone have any questions? Okay, there's one in the Q&A section. Is 3D structure helping for MTB reports if it's resolved for patients in 5-minute window? I'm not sure what the end of that means, but are they useful for molecular tumor board meetings?
So yeah, that's a good question. The 3D structure is, so this is a functionality that we have available. That I think is important, and we used it, for certain occasions, but obviously, if you show the 3D structure, then actually you talk a little bit longer than 5 minutes for the patient. So when I say, this is really like the average amount. So if there is something that we really find interesting due to the 3D structure, then actually, we will invest, we will spend more time on the patient. But definitely, from time to time, it's really important. So especially for variant of unknown significance, where we don't exactly know what the impact of the mutation is. And in those occasions, the 3D structure can really help whether a mutation could be actually pathogenic or not.
Okay, great. Mark Rubin, do you have a question?
Yeah, I had two maybe interrelated questions. So the first
Sorry,
is...
we can't actually hear you at the moment.
Can you hear me now, or hello?
Yes, yeah, we can hear you.
Hello? Hello?
We can see and hear you.
You can, okay. So the two interrelated questions. So in the PROfound trial, Foundation Medicine one was used for testing, and approximately 30% of the time the assay failed. I'm just wondering what the failure rate is in more of a daily practice, not in maybe a clinical trials practice where samples are being sent. So that's one question. The other is, you highlighted the TMPRSS2 ERG rearrangement. One of the issues is that just looking for the 3 megabase loss detects 2/3, but not all of the fusions. I'm just wondering if that's important, or if it's just, do you indicate that it's you may miss some of the balanced rearrangements?
So regarding failure rate, so yeah, we have a failure rate in our lab, but I don't know exactly how many are actually failing in the lab. So in most of the cases, it's just due to poor DNA quality, or we have just too little DNA amount actually available. So these are the two most common failure reasons. I mean, it's not 50%. It's also not 60% or 40%. It's I would say in a single-digit region. But I don't know exactly now how many, are really failing in our lab. But I would say really it's in a lower single-digit region. And regarding so the temp, yeah, I mean that's the TMPRSS fusion. So the thing is, I mean, we are aware that due to the fact that we just sequence DNA, that we can still miss a certain fusion events, or like if you so sequencing intronic regions is also not easy, because you have to have really like very well coverage throughout the entire intronic region, and introns are very large. So there can be occasions where where we miss fusion events. To be honest, I mean, so far, I mean, so far through all of the tests that we run, and then other tests that were run maybe in parallel by other groups, I mean, there was not a single occasion where actually we were notified that we missed a fusion event. But for sure, there must be a certain number of events that we missed. But yeah, but it's not RNA sequencing. So with RNA sequencing, it's kind of the gold standard for fusion detection. It's DNA sequencing, and it has problems. But yeah, but I think it's still compared to other assays, it's still doing a great job.
Okay, thanks.
Okay.